So in the previous class last week, we talked about Jablonski diagram, which is a central conceptual framework for anything that you do in fluorescence. Therefore, I would like to spend a bit more time on this particular aspect. And today I'm going to use the whiteboard that is available with my tablet. Okay. So I guess all of you can see my whiteboard. Okay. So so as I have already mentioned before, that when we talk about fluorescence, we essentially worry about the singlet excited state. And of course, as I mentioned, most of the electronic states you will worry about as are singlet excited states or singlet states where spins, electron spins are paired. However, there are of course electronic states which are triplet. Okay, so the triplet comes from the fact that if you have so spin, of course, half spin, and if you add this, you get singlet, and for triplet, of course, you have the spin, unpaired. Okay, but for most fluorescence applications, we will worry about singlet excited state. But I'll also show you a little bit of example of triplet excited state. Okay. All right. So as we have already discussed, that you have a ground state which is denoted as S naught. Okay. S again denotes the singlet state. This is, of course, a ground state. And you can have, of course, S1, which is the first electronic excited state. You can have S2, which is the second electronic excited state, S3, and so on. Okay. So each of these electronic excited states or ground state, of course, S0 is ground electronic state, are associated with much smaller energy separated vibrational states. Okay, so electronic states are much more well separated in terms of energy. So energy is, of course, along this axis, and therefore you have also vibrational states, okay, associated with each of these electronic states. Okay. In other words, molecules can, of course, vibrate, and they can populate different vibrational states within the given electronic state. And that's true for any other states that you might want to worry about. Okay. So the idea here is vibrations and any molecular movements, okay, are or atomic displacements in the molecule through vibrations, which give rise to different arrangements. Okay, that set of those processes are much slower compared to the electronic excitation. Okay, this is what is known as the Franck-Condon principle. Okay, the electronic excitation is much faster compared to any movements through vibration or even translation and other events. All right. So of course one can show, which I'm not, of course, describing this. One can show that electronic excitation is much faster. Typically, of the order of a femtosecond. Okay, and of course, as I've already mentioned, and many of you already know, that femtosecond is basically 10 to the power 15 seconds, which falls in the regime of ultra fast. Okay, and then you have got picoseconds, which is 10 raised to the power minus 12. Then you have nanosecond, microsecond, millisecond, and then second. So that's how they are separated. So, so just imagine you have a molecule, a chromophore, which can now undergo excitation from the ground electronic state to the higher electronic state. Okay, so now if you have wavelength, excitation wavelength, which matches this energy gap, then of course you can promote this to S1. Okay, or if you have higher energy photon in terms of the wavelength of light. Okay, this is basically now wavelength of light. Okay, so wavelength of light will determine where you are basically exciting. Okay, now of course excitation essentially means absorption of photon, and of course you are familiar with absorption, Lambert-Beer's law, molar extinction coefficient, and so on. Okay, so molar extinction coefficient, or epsilon, essentially quantifies the absorption probability. Okay, it is also termed as absorption cross section, so it is related to absorption cross section. So how much light a chromophore can absorb. Okay, and of course upon absorption of light, it gets excited to the higher excited state. Okay, and as I said, this particular process of excitation, either from S0 to S1 or S0 to S2, this process occurs on the femtosecond time scale. In other words, 10 to the power minus 15 seconds. Okay, which is really fast. And during this time scale, nothing moves in the molecule because vibrational processes are much slower on the picosecond time scale. Okay, so this goes up there. And of course, if it goes to the higher electronic excited state like S2, then of course there are other relaxation mechanisms by which it comes to the the first excited state. Okay, and fluorescence always occur from the first excited state, which is S1. Okay, the reason is this process, there are two or three processes, excited state processes, that one will worry about. One is, of course, this is called internal conversion. Okay, so basically from S2 to S1, okay, it comes down to the lower excited state. If you have excited it to higher excited state like S2, if you have excited to S1, of course, this process is not there. Then, of course, it relaxes here in the first excited state through vibrational relaxation. So it's called vibrational relaxation. Okay, so the vibrational relaxation occurs before emission of photons, because vibrational relaxation time scale is of the order of picoseconds. Okay, internal conversion also occurs on the time scale of picoseconds. Okay, and fluorescence occurs on a much longer time scale, that is nanoseconds. Okay, then of course it can come back to the ground state. Okay, And when it comes back to the ground state, then of course it gives you, it emits photon of this particular energy corresponding to this particular energy gap. And this is essentially fluorescence emission. Okay, this is essentially fluorescence emission. And this gives you a photon which is now different in color compared to absorption. Okay, so in absorption, of course, you might have used either this or corresponding to this energy gap. And now because of vibrational relaxation, there will be a little bit of shift in the fluorescence spectrum as compared to the absorption spectrum. Now, another thing can happen here, okay? This particular vibrationally relaxed state can now also undergo much more stabilization to here, okay? Due to the fact called solvent relaxation. Okay, and then of course, if the state comes down by solvent relaxation mechanism, then of course your photon that gets emitted is even lower in energy. Okay, therefore solvent relaxation will make it even more redshifted. And this is why you see so-called Stokes shift in fluorescence emission. Okay, so again, so you are talking about two kinds of processes. If you remove this S2 complications from S2, because in many fluorophores you don't even go to S2, you just go to S1, which is the first excited state. Then of course you've got quite a few processes. One, of course, is vibration relaxation. Okay. Now, and then solvent relaxation, which also occurs on the, let's say, tens of picoseconds. Okay. Let's say even to hundreds of picoseconds, depending on how organized the solvent molecules are, or water molecules in case of water as solvent, how organized the solvent molecules are. So it can be on the picosecond to tens of picosecond time scale. And then of course you have fluorescence emission, which occurs on the nanosecond time scale. So the question is why fluorescence occurs from the lowest vibrational state of the higher excited state. That answer lies in the time scale, because vibrational relaxation is much faster compared to fluorescence emission. Okay, therefore it has to first get relaxed in the higher excited state like S1, and then of course there is vibrational relaxation. In conjunction with that, you can also have solvent relaxation depending on the dipolar nature of the solvent. I'll come to this dipolar relaxation soon. And then of course you have fluorescence emission, which is on the nanosecond time scale. Now, if you look at the plot of, let's say, absorption and fluorescence emission, let's say this is absorption, this is, let's say here you have an absorption spectrum, okay? And then fluorescence invariably, okay, this is absorption or fluorescence, so that I can show it on the same plot, although you are plotting different things, units and everything will be different. In absorption, your unit is essentially, absorbance, whereas in fluorescence, your unit is how many photons you get as a, per unit time, let's say per second, okay? Now, if you don't have any solvent relaxation, there might be a very little shift. Okay, and now, okay, I'll just change the color um, to, let's say, this one. So I'm normalizing, of course, the maximum intensity. Okay, and these two are not in same unit. So this is absorption spectrum. This is, let's say, typical fluorescence spectrum if you were only to consider vibrational relaxation, because this much energy is lost because it has now come back to the vibrationally relaxed state, because often you excite it all these levels, because light you use, of course, we use monochromatic light. Let's say you use the lambda absorption max light. Okay. Typically, you can excite the fluorophore using the light which is at the center of the absorption spectrum. Okay. So you're using a monochromatic light, but no light is monochromatic, because even if you have a band pass of bandwidth or band pass of, let's say, 1 nanometer, you still will populate other vibrational states, because your excitation light source is not precisely monochromatic. Okay, there is always some bandwidth associated with the excitation. Therefore, it goes to any of the states, and of course, it will come back. Then there will be a shift like this, and this shift will be very little. Okay, maybe something like 10 nanometer or so, typically. Okay, depending on the fluorophore vibrational relaxation mechanisms and so forth. But if you have solvent molecules around it, now why should you have solvent relaxation? As I've already mentioned, that in the higher excited state, so in the let's say in the ground state here, you have certain dipole moment, or let's say no dipole moment, and solvent molecules are organized in some fashion to stabilize this particular, let's say, del positive, del negative, and so on. Let's say oxygen here, hydrogen here, or vice versa, depending on the exact nature of the dipole. But upon excitation, you've created an instantaneous dipole moment, which is much more, first of all, or no dipole to high dipole moment. And the solvent molecules have not moved around, because Frank Condon principle already says that nothing moves around except electronic excitation. Therefore, the dipole moment at the excited state is now very different, both in terms of the direction and the magnitude of the dipole moment. Therefore, this state is an unrelaxed state. Now, what water molecules have to basically move around, translate, rotate, or solvent molecules have to translate and rotate to stabilize it to the solvent relaxed state. Okay, so you need to consider the mobility of solvent molecules, okay, in response to instantaneously created dipole, which is now an high energy species, because it is not, the solvent molecules are not able to solvate the newly created dipole upon excitation, right? Therefore, the water molecules have to move around. And of course, during this time, there is also vibrational relaxation happening. So all these things are sort of happening on the picosecond time scale, okay? Picosecond to tens of picoseconds time scale. And of course, after water molecules or solvent molecules are able to completely solvent this, then you have a solvent relaxed state. And upon solvent relaxation, there is also a drop in the energy as depicted here. Therefore, you are now going to see a much more red-shifted emission, okay? And this is due to now, this is due to just vibrational relaxation. Now this is due to both vibrational plus solvent relaxation, okay? So there is a shift in the fluorescence spectrum compared to the absorption max. Therefore, what essentially you are talking about is the Stokes shift, okay? Because it was discovered first by Stokes, okay? So the name is Stokes, therefore Stokes shift, okay, equals to essentially lambda emission max, okay, minus lambda absorption max, or lambda max, okay. So this is basically Stokes shift. Now this is a very important parameter when you are doing fluorescence spectroscopy, even microscopy, okay. And this is something which is basically the most important thing in fluorescence, okay. So let me also tell you something about the what you call as fluorescence quantum yield, which is basically the efficiency of fluorescence, okay. Now if you use, let's say, 100 photons, let's say a molecule absorbed 100 photons. Now the question you are asking, are you getting all 100 photons back? And of course, they will be distributed in terms of this wavelength distribution, okay. So are you going to get all 100 photons back? The answer is no. You are going to get only a fraction of photons you have used for excitation. And this is what is known as the quantum yield, okay. Now we will come back to quantum yield again tomorrow, okay. But just to tell you that quantum yield essentially tells you the efficiencies, quantifies the efficiency of fluorescence. So if you have got a chromophore which has a certain absorption cross section, so which is capable of absorbing this many number of photons, let's say per unit time or per so are you going to get all the photons returned to you as fluorescent photons, okay. And the fraction is basically termed as quantum yield. So you can imagine that, let's say if you have 90% quantum yield or 0.9 quantum yield, depending on how you represent it. So if then, of course, 10 photons have been lost in some place, where did they go, right? So if 100 photons have been absorbed, ideally you should get 100 photons back, right, in fluorescence after all these processes. But then in the best case scenario, you can possibly get about 94 to 95% of photons back as fluorescent photons, okay. And for many fluorophores, even 90% photons are lost. You get only 10% fluorescence, which is the quantum yield will be 0.1 in that case. So now, why do you lose photons? Because if you have used the photons for excitation, then of course you should get all the photons back. And the fluorescence efficiency is basically described as quantum yield, okay. The reason you lose photons, in addition to the processes that I have just described, there is also non-radiative process, okay, or a set of non-radiative processes, okay. So what I've shown over here using Jablonski diagram is essentially the radiative channel, okay. The molecule gets excited, typically to S1, then there is vibrational relaxation, then of course there are cases where you can excite it to S2 and then internal conversion, then vibrational relaxation, followed by solvent relaxation. They are by and large, overlapping time zone, but vibrational relaxation is a bit faster compared to solvent relaxation. And then, of course, you get the photon back after, let's say, nanosecond, which is what is termed as the fluorescence lifetime of a particular chromophore. Okay. Now, this is basically the radiative pathway. And now there is also a non-radiative pathway. What happens is this excited state dipole, which is shown over here, can also the energy can get dissipated by interacting with the solvent molecules around it. Okay. Therefore, what can happen is called thermal dissipation. Okay. So the higher excited state, chromophore can now interact, collide with the solvent. Of course, solvent molecules are, going around Brownian motion, and then they collide with the excited state dipole. Therefore, the energy gets dissipated through the solvent molecules in terms of thermal fluctuations. And then there is, gradual drop of this excited state to the ground state. Okay. Therefore, it is through collision with the solvent molecules. Okay. And this is what is known as non-radiative process, one of the non-radiative processes. There are other non-radiative processes we will talk about, called fluorescence quenching, fluorescence resonance energy transfer, and so forth. But solvent molecules alone can interact with the fluorophore and de-excite it by just energy dissipation through non-radiative process. So that's where part of the excited state is decaying in the non-radiative channel. So you have radiative and non-radiative. Okay. Now, so you've now got two rate constants. Okay, it's like a chemical reaction. You have it is sort of like what you call as parallel reaction. Okay, not really consecutive. It's a parallel reaction, so you can have radiative decay, non-radiative decay, depending on the rate constant of radiative decay, which is, let's say, Kr, and non-radiative, which is Knr. You will have basically see this interplay of Kr and Knr will give you the net quantum yield. If Knr is highly operative, highly efficient, much faster compared to Kr, then of course you are going to see primarily non-radiative decay. In that case, your fluorescence quantum yield is going to be much lower. Okay, whereas non-radiative decay channel, when it is almost shut off, then of course you get primarily radiative decay. Therefore, you will get very high fluorescence quantum yield, close to 95% or so. Now, what's the cause of this non-radiative decay? The cause of non-radiative decay, of course, multifold, but one major cause is, of course, its interaction, the fluorescence excited states interaction with the solvent molecules. Okay. And when this interaction is going to be more profound, obviously, if you have changed the dipole moment in the excited state, so if there is a significant change in the dipole moment in the excited state, then of course water molecules have to sort of reorient, to stabilize the excited state dipole. Therefore, under that particular condition, you are expected a lot of collision with the excited state and the solvent molecules. Therefore, if you have significant solvent relaxation, you can also expect a significant non-radiative decay channel. Okay. Therefore, if so, in other words, if you can sort of go back to the structures and of the fluorophores, so if you excite and upon excitation, if there is a drastic change in the direction and the magnitude of the dipole moment in the excited state, then of course there will be a very strong solvent relaxation component, by which there will be which will be evident by a significant red shift. Okay, this is red shift. Okay, in from the absorption to fluorescence. Okay, absorption max to the fluorescence max. Okay, there are fluorophores which can give you a shift of 100 or more, 150 nanometer fluorescence shift. Okay, and of course those are the fluorophores which will have much lower quantum yield. They are much less efficient fluorophore because they interact in the excited state, they interact with the solvent or water in the vast majority of cases, water. Since they interact and get stabilized by the solvent relaxation channel, they are also prone to get dissipated without emitting a photon, which will give you fluorescence. Therefore, fluorescence quantum yield drops drastically. Okay. Now, this is again very important. Sort of, I'm trying to connect a lot of things together. Of course, if you think we need to discuss again tomorrow a little bit in the early part of our class, we can do that, because I'm trying to combine a lot of things because they are all related, first of all. And this is the single most important thing in fluorescence spectroscopy and microscopy. Any fluorescence application, you need to know this, okay? The origin of Stokes shift. And of course, you can use the, all this information to get somewhat, quantitative or semi-quantitative feeling of the environment around the fluorophore. Okay, so why do I say this? So the entire thing that I've now talked about, that the fluorescence Stokes shift, and of course much lower, of course, if you have a lower fluorescence quantum yield, then of course your fluorescence will not be that high, it'll be much weaker fluorescence. Okay, so we will come back to quantum yield later. So you have much lower fluorescence intensity as opposed to what you're supposed to see, based on the absorption or absorbance. Okay, now why these things are so important? So you can imagine the entire process. Okay, the cycle, of course it's a cycle, because from S0 you go to S1, gets relaxed in the vibrational excited states, and then solvent relaxation, then it comes down, then again it can go up, and so on and so forth, right? So the entire process is dependent on two critical things. One, of course, is the nature of the chromophore or fluorophore. If, of course, a chromophore emits, we term this fluorophore, right? So entire thing is dependent on two things. One, of course, is the fluorophore, the quantum chemical behavior of the fluorophore, okay, which is what is important, and the solvent molecules or surroundings. I mean, you can also make it a little bit broader to say the immediate vicinity of the fluorophore. And that's the reason fluorescence has become so powerful, because it can now sense the immediate vicinity, the local micro environment around the fluorophore. Okay, it could be solvent, it could be dipolar medium, because most solvents are dipolar, like water, like DMSO, like methanol, ethanol, and so forth. And now, if you introduce this fluorophore, let's say in a hydrocarbon solvent, okay, let's say paraffinol or hexane, okay, great example. Then, of course, the solvent is no longer dipole, okay, it is a hydrocarbon solvent, okay, and the process that I have described right here, okay, the solvent relaxation is not going to take place, because no matter how many solvent molecules want to tumble around, translate around to stabilize the dipole, they will not be able to stabilize the dipole, because the intrinsic nature of solvent molecules dictate that they don't stabilize a dipole, right? Because they are not dipolar, right? Therefore, they won't be able to stabilize the dipole, right? Therefore, the solvent relaxation step will not be there for hydrocarbons. Let's say for hydrocarbon solvent like hexane, there will be no solvent relaxation. Therefore, your fluorescence Stokes shift that you were supposed to see for water as a solvent, you will not see for hexane. So there will be much less fluorescence Stokes shift, and the Stokes shift will be only due to the vibrational relaxation, which is much smaller, right? Therefore, by looking at the fluorescence spectrum, you will be able to make an assessment what solvent this is, or what kind of nature the solvent has. Okay, so if there is lack of solvent relaxation, because there is no solvent relaxation in a hydrocarbon solvent, then of course you also don't lose fluorescence, because it doesn't extensively interact with the dipolar medium to dissipate the energy. So non-radiative process will also be less efficient for hydrocarbon. Therefore, you are going to see much higher fluorescence in hydrocarbon or in a hydrophobic environment. Of course, in biology, you don't use hydrocarbon, but there is hydrophobic environment in lipids and membranes and proteins and so on. Therefore, in the presence of hydrocarbon solvent or hydrophobic or lipophilic solvent, you are not going to see extensive non-radiative decay. Therefore, your decay will be primarily governed by radiative decay. Therefore, you'll have a longer fluorescence lifetime and higher fluorescence intensity and lower Stokes shift or smaller Stokes shift. So these things will go hand in hand. Therefore, if you use a given chromophore, okay, you will be able to make an assessment of the immediate vicinity or the local environment of the fluorophore. Okay. So let me also summarize because I've talked about quite a few different things, and then I will come back to you if you have questions. So, so, so the first step of fluorescence, of course, is absorption of photons. And all of you know, I mean, I've not discussed absorption in so much details, assuming that all of you have studied absorption at some point, and you know absorbance. Basically, you can plot absorbance versus concentration, and you get something like this, which is known as Lambert-Beer's relationship. And of course, the slope basically gives you the molar extension coefficient, because A equals E, C, and L is the path length of the cell where you're studying, recording your absorbance. And of course, beyond a certain point, there is some non-linearity, and of course, you know why that happens. So you need to be in the linear regime, and epsilon essentially is it characterizes the particular chromophore, how much photon it can absorb, which is expressed in terms of mole inverse, centimeter inverse. Okay, so so that tells you how much light this can basically absorb, and and of course, normally you use absorption max to excite. In fluorescence, of course, you can use, away from the absorption max, you can also use wavelength, which is off peak wavelengths. And excitation, of course, take the takes the excited state from ground state to the first electronic excited state typically, but there could also be, it can also go to the higher excited state if you are going in the blue end of the spectrum. And then, of course, there are relaxation processes at the excited state, like internal conversion, vibrational relaxation, solvent relaxation, which typically occur on the picosecond to tenths of picoseconds time scale. And then, of course, there is fluorescence emission, which is much more delayed compared to the absorption. Typically occurs on the nanosecond time scale. But of course, fluorescence lifetime is a convolution of the radiative and non-radiative processes, because it has both radiative and non-radiative. So observed fluorescence lifetime has both of this. And of course, if non-radiative process is highly efficient, then of course fluorescence lifetime is going to be much shorter. And as a consequence, you're going to see much more red shift from the absorption spectrum. So there is much more loss in the energy because of solvent relaxation, extensive solvation at the excited state. And that'll also tell you that due to non-radiative process, there'll be fluorescence will be lost due to non-radiative processes. Therefore, fluorescence intensity will drop, and therefore quantum yield will be much lower. Okay, as opposed to this in dipolar solvent, if you go to hydrocarbon or more hydrophobic environment, you are expected to see much more fluorescence, lower Stokes shift or smaller Stokes shift, and longer fluorescence lifetime, because non-radiative process is much less efficient in the hydrocarbon solvent. Okay. Therefore, in summary, fluorescence behavior is dependent not only on the chromophore or the fluorophore, but also on the solvent environment or the immediate vicinity. Therefore, fluorescence allows you to record the environment sensitive information uh, using a variety of this readouts like Stokes shift, fluorescence lifetime, quantum yield, and so forth. Okay. So I would like to stop here, and then if you have questions, we can also discuss.